Hello, everyone. I'm Dr. Kobenan Koffi Christophe. I'm from Ivory Coast, West Côte d'Ivoire. I'm agrophysiologist and entomologist. My presentation this afternoon is chemical composition, antioxidant activity, cholinesterase inhibitor, and in vitro insecticidal potentiality of essential oils of Lippia multiflora and Eucalyptus globulus on the main carpophagous pests on cotton plants in Côte d'Ivoire. Please excuse me very much. I'm not an Anglophone, I'm more for Francophone, but I will make an effort to present my research work to you. It's worth noting that cotton cultivation repeatedly and abusively use numerous synthetic insecticides. These are serious consequences for the health of cotton growers and for the environment. In order to limit the use of synthetic chemical insecticides to growers, the general aims of the study is to identify biologically effective botanical insecticides that present less risk to the environment and human health. Especially, the aim was firstly to determine the insecticidal potentiality of essential oil on the main carpophagous insects of cotton, and secondly, to determine the chemical composition and biological activity of essential oils. The study was carried out in the town of Boaké in Côte d'Ivoire. The experiments were carried out in the agricultural entomology laboratory of the Centre National of Research Agronomic, more precisely at the Cotton Research Station. Two aromatic plants were used. The first plant on the far left of the slide is called Eucalyptus globulus, and the second is Lippia multiflora. These plants are very common in north of my country. The plants we have just seen were dry for 1 to 2 weeks, and we used a clevenger to extract the essential oils. 3 main carpophagous insects were used to toxicological testing. These are Pectinophora possipiella, Tomatotibia leucotreta, and Helicoverpa amigera. It should be noted that the larvae of these insects have a very negative impact of cotton yields as they attack the cotton bolls, contains the fiber. The slides show how we performed the toxicological tests. In fact, we carry out the test by topical application. This means that we applied a drop of the dopper solution to a bath of 10 insects at a precise concentration of the essence dorsal side, and it was repeated three times. It should be noted that for each essential oils, we prepared a stock solution, and from the stock solution, we prepared 9 daughter solution concentrated from 0.15 to 64. Then 48 hours later, we calculate mortality and use Windell software to calculate lethal concentration, in particular the LC50. The LC50 is concentration required to cause the death of 50 of the insect tested. Note that lower the LC50, the more toxic the product. Here we have presented an overview of the work we carried out in our trial. Overall, we collected leaves from 2 local plants, Lippia and Eucalyptus, we then extract the essential oils. Once the essential oils had been obtained, we carry out a chemical analysis, use chromatography to determine the chemical composition. In addition to this chemical analysis, we evaluate the antioxidant and enzymatic activity. After chemical analysis, we evaluated the insecticidal efficacy on free main cotton pests in the laboratory. About the result obtained, the slides show the chemical composition of 2 essential oils we evaluated. Chromatography revealed the following main chemical elements in Eucalyptus globulus essential contains alpha-pinene, limonene, cimene, 1.8 cineol or eucalyptol, and in Lippia multiflora we have alpha-philandrine, P-cinem, 1.8 cineol or eucalyptol, geraniol, and meral. About the biological activity of essential oils, Lippia multiflora essential oils have better antioxidant activity than Eucalyptus globulus, with a DPPH value 7.05. With regard to cholinesterase activity, the 2 essential oils are not dissimilar. On the other hand, Lippia multiflora essential oils has very good tyrosinase activity. After evaluating the insecticidal activity of essential oils, we observed that Lippia multiflora essential oils was the most toxic on Pectinophora goxypiela. In fact, here on this graph, you can see that these essential oils has a very low lethal concentration, whereas Eucalyptus oil has high lethal concentration. On the second insect tested, Lippia multiflora essential oils was again the most toxic with a concentration of 1.39%. On Helicoverpa amigera, Lippia multiflora essential oils was most toxic. In fact, we can see here that the lethal concentration these essential oils was very low at 7.2%. In conclusion, essential oils, notably Lippia multiflora and Eucalyptus globulus, have vastly variable chemical composition and have excellent enzymatic and antioxidant activity. The study also revealed that Lippia multiflora essential oils has very good insecticidal against the main carpophagous insects, namely Pectinophora goxypiela, Tomatotibia leucotreta, and Helicoverpa amigera. The essential oils of Lippia multiflora appeared promising for the development of the bioinsecticide for phytosanitary protection of cottons. However, it would be important to evaluate the efficacy of Lippia essential in the greenhouse and in the field. it will be very difficult to me to answer your question orally. However, if you have any concern about my work, you can send me an email and I'll be able to send you all the answers. Thank you for the kind attention. Thank you all very much.